I will turn it over to Dr. Paul Offit. You all met him in August. He is the director of the Vaccine Education Center and an attending physician in the Division of Infectious Diseases at the Children's Hospital of Philadelphia. He's a professor at the Perelman School of Medicine at the University of Pennsylvania, and he's a member of VRBPAC, the FDA's Vaccine and Related Biologics Advisory Committee. Dr. Offit has obviously been in the news lately, and it's been a very newsy week in the last week. So I'm just to turn it over to him for some quick general thoughts about kind of where the vaccine process stands, and then this is mostly going to be Q&A. So I'll turn it over to Dr. Offit. I want to thank you very much again, Dr. Offit, for coming and joining us. Looking forward to hearing from you, and then we'll get to as many questions as we can.
Okay, so I'll try and do this all in about 5 minutes, but let me sort of contrast my experience with developing a vaccine with the current experience with developing a vaccine. So I was part of a team at Children's Hospital of Philadelphia that created the strains that became the bovine human reassortant vaccine Rototec. The way that worked was I worked in a laboratory for about 10 years, working with experimental animals, trying to figure out what part of the virus made you sick, what part of the virus induced a protective immune response. We then created these strains that we thought could do the best of both worlds, induce an immune response without making you sick. We then did a series of phase 1. That took 10 years. We then did a series of phase 1, phase 2, and phase 3 trials in collaboration with Merck, which took about 16 years, ending in a phase 3 trial. A placebo-controlled, prospective, 70,000 infant, 11 country, 4-year, $350 million trial to prove that the vaccine was what we hoped it would be, which was safe and effective. We then submitted those data to the FDA. If Penny Heaton, who was in charge of the program at the time, said if we took all the clinical material that we had and lined it up, sort of stacked it up page on top of page on top of page, it would exceed the height of the Sears Tower. I mean, that's how much information is generated from a phase 3 trial. You then submit it to the FDA. If it has its typical review at the time, and this was in the mid-2000s, if it had a typical review, it would be a year to review those data. You can get an expedited review for an unmet medical need in 6 months. That's typical. Then the FDA then turns it over to the VRBPAC committee. The VRBPAC committee reviews the data, and they reviewed that data and then made a recommendation, and then licensed the vaccine on February the 6th. 2 weeks later, the Advisory Committee for Immunization Practices recommended it. So that was it. That was a 26-year effort, which was just summarized, by the way, in about a minute and a half. It's depressing. You can actually take your entire career and summarize it in that short a period of time. That was that. So how does that differ from this? The size of the trials are the same. I mean, when you see Moderna does a 30,000 person trial, Pfizer a 44,000 person trial, Johnson & Johnson a 60,000 person trial, that is the size of a trial of any pediatric vaccine. The difference is that when they submit it to the FDA, they're not submitting it as a biological license application for licensure. They're submitting it for approval through emergency use authorization. But you could argue if it's the same size of a trial as any other pediatric vaccine, why do it through an EUA? Why not just do it as a licensed agreement? And the reason is the length of the trial. Although the size is right, the length that these Moderna and Pfizer trials start at the end of July. You're only going to know whether or not these vaccines are effective for a couple months by the time they get approved. The FDA would, I think, be very unlikely to approve something that had not been shown to be effective for longer than that. On the other hand, you're not going to do a 2 or 3 year trial. I mean, our trial, the rotavirus vaccine trial, was a 4-year study. The human papillomavirus vaccine trial, which was a 30,000 person trial, was a 7-year study. You're not going to do that kind of study when you have 250,000+ people dying this year. So then the question becomes, and this was our last, the October 22nd VRBPAC meeting, are we comfortable with an EUA? As distinct from a license. I mean, what is it about this makes us comfortable or uncomfortable? That really was the subtext, I think, of that meeting. Are we comfortable with an EUA? The same EUA process that allowed hydroxychloroquine to be used, which didn't work to treat or prevent disease. I mean, the same EUA process that worked or that was used to give us convalescent plasma, which certainly hadn't been shown to work. And it was clear at that point that it seemed that the administration was influencing the FDA's decision, that the FDA was serving more at the whim of administration than doing its job, which is to protect the American public from products that are unsafe or ineffective or both. But that's changed now. I really do think that when the FDA said we want to see safety data at least 2 months after dose 2 for at least half of those who got vaccines, that was, I think, Commissioner Hahn standing up to the administration saying we need 2 months of safety data after the last dose. So we're going to be, the FDA is going to be looking at these data. They'll probably look, instead of for a year or 6 months, they'll be looking at reams of data for a couple of weeks, and then we will probably see this sometime in the second week of December. So are we reasonably reducing a degree of uncertainty with this vaccine with which we can be comfortable, either from a safety or efficacy standpoint? So we'll know that this vaccine, if you look at the latest Pfizer press release, they told you basically that it was effective at roughly the 95% level 1 week after dose 2. That's when you have your peak immune response. I mean, so really what you would like to know is, was it also effective a month later after dose 2 or 2 months later after dose 2, for which they have data? I mean, because they have the safety data. So there's no reason that you wouldn't expect they would also have the efficacy data. It'd be nice to have all 3 time points: 7 days after dose 2, 1 month after dose 2, 2 months after dose 2. Is there any fraying of immunity during that period of time? Because that's a fear. Are we going to have a vaccine that's effective for much shorter than we thought, which would be a problem? Now, I don't think that's going to be a problem, but you don't know. So you have 2 months of data, and then we'll look to see whether it's effective six months later, or 9 months later, or a year later. But I think if it is effective for that period of time, it's likely to be for 2 months. It's likely to be effective for longer. All that really depends on the ability of these vaccines to induce high frequencies of memory B and T cells. I mean, people always worry when you see antibody titers decline. That's not what worries me. What worries me is that you don't have a high frequency initially of memory B and T cells, because this is not really a short incubation period disease. It has an incubation period of about 6 days, which means that when you're exposed, then re-exposed to the virus, or you're vaccinated, then you're exposed, you really have a few days to have these memory cells become activated and differentiate to become, say, from a B cell standpoint, antibody secreting cells. That's enough time to modify illness. So we'll see. And then in terms of safety, and I've talked to we've had a call with the CDC, our Vaccine Education Center, not long ago, to ask the question, if you look at the serious side effects associated with vaccines, to what extent are you going to pick them up within 2 months? And I think the answer is pretty much, for the ones that we've seen so far in the past 2 centuries of having vaccines, I think you'll pick most of them up. I mean, so for example, polio caused by the oral polio vaccine, which occurred in 2.4 million doses, all occurred obviously within 6 weeks. Guillain-Barré syndrome following influenza vaccine. Narcolepsy, a disorder of wakefulness following the squalene adjuvant flu vaccine in Europe. Obviously, latex or gelatin allergies associated with vaccines, which are immediate. The viscerotropic disease following yellow fever vaccine, or otherwise known as yellow fever following yellow fever vaccine, would all be picked up quickly, so meaning within 6 weeks. So I think the 2-month safety net at least tells you that you don't have a serious adverse event that is relatively uncommon. It doesn't tell you whether you don't have a rare serious adverse event, but that's true for any medical product. 20,000 people isn't 20 million people, so you'll know that later. But I think we have with this, we'll see what the data look like. But assuming the top of line data that you saw in the press release is confirmed by when you look at all the data, I think that we'll have reduced a critical amount of uncertainty. But you never know, and so you have to be humble. Keep your eyes open, and realize that you may find things that you didn't expect. And just from my personal standpoint, I mean, I worked on rotaviruses for 26 years, and our vaccine came out in 2006. But there was a vaccine that came out before us, RotaShield, that came out in the late 1990s, that was found to be a rare cause of intussusception or intestinal blockage. Now, at the time that was found, the late 1990s, that virus had been studied for 50 years. I mean, in experimental animals and people, that was a well-known pathogen that affects pretty much all mammals on this planet. So veterinarians had been studying it, clinicians had been studying it, and nonetheless, despite 50 years of study, that finding of intussusception was a complete surprise. This virus has been studied for a year, it's already had a number of clinical and pathological surprises. So I do think that we can't be so sure of ourselves that there's not going to be a problem that we haven't anticipated. See my little dog back there? See my dog walking behind me?
Oh, yeah.
That's her. See her?
What's the dog's name?
Rosie. She has a SAG card, a Screen Actors Guild card. So we're good. Don't worry about it.
Okay.
So that's it. That would be my opening statement, and then it's all yours. And Rose also is here to answer any questions that you want.
Okay, let's go to Lisa first. And Lisa, give Dr. Offit a reminder of who you work for.
Yeah, hi, Dr. Offit. I'm Lisa Krieger at the San Jose Mercury News. Your grand rounds at UCSF yesterday was stellar, so thank you. First, two questions. One is, at the VRPAC meeting, what will you be looking for in terms of data that we haven't learned from the press release? Do you have outstanding questions that were not addressed by the press release? And then the second question is, how do we learn about transmissibility of the virus in vaccinated people? Are there any trials ongoing? Do these trials address that at all?
Right.
Those are my two questions.
Sure. So I'll take the first one first. I think that you'd like to see, if at all, you can get more than 1 point on the curve here in terms of effectiveness. Because in theory, they should have a reasonable amount of data 2 months after the dose 2. So is there any evidence that effectiveness wanes? What kind of evidence do they have at all, if at all, of sort of frequency of memory B and T cells? That would be really important for me to know that I mean, when you're looking at antibody responses, virus-specific neutralizing antibody responses, that's really the tip of the iceberg. You want to make sure that there's a big iceberg below it, or the area below the water. And then obviously the sort of subset analysis. I mean, let's break down the data on people who are greater than 65. What do they know about people who either were or were not infected prior to entering the trial? But I think Moderna didn't distinguish those, but I think Pfizer might have. I think Pfizer might have said we're not going to include people who've already have antibodies, so we wouldn't have those data from them, but would for Moderna. But we'll see. Do they have any information on people who have diabetes, people who are obese? And then really the group you always learn the most from are the people who got vaccinated but weren't protected. That's really the group that gives you the most information. I mean, why weren't they? Did they not have a neutralizing antibody response as compared to others? Was it lesser than others? Did they have certain medical conditions? Were they older? What was there anything that could explain that? That's really where you learn. And that's if you're going to find an immunological correlative protection, that's often where you find it, in those people who got the vaccine but weren't protected. So at least that's some of the things that come to mind. In terms of the other question, which is really important, is are we looking in these two trials, at least the two mRNA vaccine trials, at just protection against illness, or do we have any information on protection against infection, during which you might still shed virus at a level that would make you contagious? My understanding is that for Pfizer, Moderna, Johnson & Johnson, AstraZeneca, those trials are not set up that way. I mean, I was on an NIH, this is recorded. Let me just say, there's an interest in doing a trial in college students to look at this issue of asymptomatic infection, and not just asymptomatic infection, whether the vaccine can protect asymptomatic infection, but if it doesn't protect asymptomatic infection, can we discern the amount of infectious virus that's being excreted or secreted, and then make a statement about whether or not we think that person would be contagious. The reason that's relevant, obviously, is let's suppose it's 95% effective at preventing disease, but only 15% effective at preventing asymptomatic infection, in which case then you may still be dangerous to others. As far as you're concerned, the person's concerned, they can think, great, I don't have a disease, I'm not gonna have to go to the hospital, I'm not gonna have to go to the morgue, I'm good. But if you care about others, and we certainly have a large swath of people in this country who don't, but I mean, if you care about others, you should still wanna wear a mask and social distance, because until we get this virus under control, that would still be a problem.
Where should we go to learn more about that trial, or can you say where that trial is?
Larry Corey.
Thanks.
University of Washington. He's the one. Well, I'll say it. I mean, at the NIH Active Group, we did talk about this when we met a couple of days ago. It was Wednesday. But he outlined that trial, and he outlined where he wanted to do that trial, and specifically which college campus. And I think it's a matter of getting the money to do the trial. I think right now it's a proposal without funding.
Got it.
Okay, next question. Let's go to Angeli.
Thank you, Chris. Good to talk to you again, Dr. Offit. Angela Kimlani from Yahoo Finance. Question, at least I actually took one of my questions, so thank you, Lisa. But one of the other questions I had was, as it stands right now, we know that one of the concerns, of course, with the trials is people either leaving or, any kind of impact to the placebo group, as well as giving them the dose once it's authorized. So I just want to know what your thoughts are on that point specifically. And then the second question that I had also was, we know a little bit more about this virus now and the vaccine's ability to respond to it. What don't we still know, and what are you waiting to learn still?
I'm sorry, say this, the last thing again.
What are you still waiting to learn as it relates to the vaccine and its ability to attack the virus right now?
All right, so a couple of things. It reminds me of one thing I didn't say that could have answered also with Lisa. The one thing that worried me a little bit looking at these trials, both Moderna and Pfizer, was that it seemed that the attack rate in the placebo group was a little lower than one would have predicted. And this could be an example of what's called volunteer bias, which is that someone who volunteers for a trial may be the kind of person who is more attentive to their health, may be the kind of person who's more likely to wear a mask and social distance, which means that they may be the kind of person who, when they're exposed to virus, would be exposed to a lesser amount of virus. And because there is an inoculum effect, which is to say the greater amount of virus you're exposed to, the more likely you are to develop moderate to severe disease, are we really looking at a vaccine that protects against a relatively low inoculum? That's also important, because it's always true that when vaccines are tested in experimental situations like these, you tend to have much higher effectiveness than when you put it out into the real world for a lot of reasons. But that would be one here, so you'd want to know that. In terms of your question, which is a hard question, Angela, I mean, so let's see, when the FDA, let's say, approves a product through emergency use authorization, as Gina, whose last name I can never remember, Gina last name that was German, Gina, it's Gina, not Gina, the person who did the presentation last time at I can never remember her name, Marion Gruber, see, Gina, Marion Gruber, who's German, has a German accent. Which she made it very clear that when we approve when we permit use of a vaccine, because that's the word, permit use of a vaccine through EUA, that's no different than allowing an investigational drug to be used. There is no ethical responsibility to give anything to a placebo group under that auspices. However, when it goes to the ACIP, the language changes. Now it's not permission, it's recommendation. And recommendation arguably establishes a standard of care. If you say that if the ACIP says, I think that Moderna's vaccine, or the Pfizer mRNA vaccine has been shown to be effective in people over 65, can you reasonably deny that to someone who's over 65? I mean, it's now a standard. And I think, they try to sort of have a middle ground with this expanded use or extended use. I just don't think that's practical. It's sort of on a case-by-case basis. You're talking about the American public. I just don't see how that plays out that way. So then the issue becomes, if that's true, then can you do placebo-controlled trials in the United States anymore if you've established a standard for Americans? In which case, then it may be that you're doing trials in other countries. And so for example, obviously Pfizer and Moderna's vaccine are not going to be available everywhere. Where they're not available, you could argue you could do those kinds of trials, which then could sort of favor arguably one company over another. I mean, if you take, say, the two companies that are making purified protein vaccines, Sanofi in collaboration with GSK and then Novartis. Sanofi, GSK are huge international companies. They could easily do studies outside of this country, whereas Novavax, which is a small Gaithersburg, Maryland company, may have trouble doing that. So that's that. But there may have been another question in there you had that I didn't answer. It's nice seeing you, Brian. I've talked to you a few times, but I've never seen your face. It was fun seeing people.
I know, it's nice to make that connection, right?
People in the news organization have faces. They're real people.
Oh, they do. And we're real people. We actually like being wallflowers mostly, but some of us are forced on air. Yeah, the second question was, what are we still waiting to learn? Either whether it comes to the virus itself or the vaccine's response. Either way, I'll take an answer.
I mean, the big issue is going to be the virus has only been out for a year. It's a single-stranded RNA virus. So like any single-stranded RNA virus, it mutates. It mutates every time it reproduces itself. I mean, RNA viruses are notoriously unfaithful. You will, if you look at if you're infected with this virus and then I catch it from you and you sequence those two strains, they won't be identical. So that's true of measles too. And so is this going to be more like flu, which mutates so much from one year to the next that it mutates away from the vaccine? Or is it going to be like measles and mumps, which, we had a measles vaccine in 1963. That's a single-stranded RNA virus, and it hasn't in any sense mutated away from the vaccine. Which of the two it will be, we'll see. There's no evidence to date, no solid evidence to date that it has mutated away from the vaccine, from what we would expect, looking at the receptor binding domain.
Thank you.
Next question goes to Andrew Dunn.
Hi, Dr. Offit, Andrew Dunn from Business Insider. I have kind of a crystal ball type question for you, but this is the one that I think friends and family ask me the most about, what this all means and how this all plays out. So I want to hear from someone much smarter than myself. What's your best guess, given what we know today, all the uncertainties, but on when the US will actually return to a level of normal in society? And can you go through what needs to happen to reach that point, not just in terms of vaccines, but therapeutics, testing, contact tracing, and so on?
So I think if you had to make a guess, and I'm willing to, make any prediction you want, as long as you don't hold me to it. See, this is the problem with recording this, but there are things that are three things that are going to work against the virus over the next year. One, a vaccine. Two, hygienic measures. And three, to some extent, although this obviously is not a strategy for elimination, more and more people have been infected. Those people who are infected are less likely to be moderately severely reinfected. That's true. And the numbers that we estimate about 11 million people have been infected, but those are people that have just been tested and found to be infected. That's obviously just a smaller part of it. And if you do the antibody studies, that's probably off by a factor of 5. I think it's probably closer to 50 million people who've been infected already. So so you've taken some people out of the population now who are likely to be infected to the point that they would have moderate to severe disease. You're going to now, the vaccine will start to roll out and you have hygienic measures to the degree that we use them. So the prediction then would be, the logical prediction would be that over time you will see a lesser and lesser number of cases, fewer and fewer cases, and then you would see fewer hospitalizations, fewer ICU admissions, and fewer deaths. So then the question is, when do you get to the point where you say, I'm comfortable going about my normal business, knowing that I could still be infected and still die from this virus? Because that's true with flu. I mean, flu last year, or this past year, caused about 400,000 hospitalizations and 20,000 deaths. I mean, the year before it was about 700,000 hospitalizations and about 60,000 deaths. We live with that every year. Now, if we wore masks and social distance every winter, would we dramatically reduce the incidence of flu in addition to the vaccine? Yes. I mean, first of all, the vaccine is not great. The vaccine is about 50% effective. And in people my age, who are over 65, much less effective. We live with that. I mean, if I go to, I'm a Philadelphia Eagles season ticket holder. I go to a Philadelphia Eagles game and stand among 67,000 screaming fans. If I wore a mask, I would probably lessen my risk of flu. Of course, it's hard to boo through a mask. That's one of the problems. But but I accept that level of risk. I've sort of grandfathered in that level of risk. I drive. I have a car. There's 32,000 people that die on the highways every year. So we do accept some level of risk, and I don't know what that is. I don't know with this what that is. I mean, right now we're just scared because it's a bat coronavirus. It's new, and also this is a scary virus. I mean, this isn't flu. Flu, either you die or you get better. I mean, this virus causes vasculitis, so it really causes more long-term problems. It can cause strokes and heart attacks and kidney disease and liver disease. What respiratory virus does that? This is a weird virus, and it should scare people. It should scare people because it can kill you, but it also should scare you because it does things to your vasculature that no respiratory virus has ever done before.
So just to follow up quickly on the timing, I've heard people talk about summer 2021 as a realistic, kind of rosy, but a realistic hope to return to normal. Is that a false hope in your mind, or do you think that's realistic?
I think it depends in large part about how much vaccine we can get out there. Assuming that we'll continue to be still pretty bad about hygienic measures, how much vaccine we get out there, how willing the population will be to take it. I think, and this seems to be counterintuitive when people look at these, these polls about how much people would want to take this vaccine. I think the vaccine is going to roll out into healthcare workers. It's going to roll out into older people. It's going to be the Beanie Baby phenomenon. I mean, this is a limited edition vaccine, which I think will make it at some level something that's going to be desirous. And then 100,000 people have been vaccinated. Then 1 million people have been vaccinated. Then 2 million people have been vaccinated. And there's no, let's assume there's no serious side effect problem. And, people feel great. And then you want it to. You want to be immune to. And I think people are going to gradually be more likely to take it if the safety issues hold up. But I do think it's just going to be a Beanie Baby thing.
Okay, next question to Kira.
Hi, Dr. Offit. Kira from Leaps Mag. Thanks for coming back to talk to us. So I know there's been a lot of discussion about kids and testing, and I just wanted to get your take on your best estimate for when kids under 12 will be included in trials and when eventually vaccine might roll out to young school children.
Okay, so you should talk to Jeff Gerber at Children's Hospital of Philadelphia, who at least emailed me recently to say that he has now been tabbed as one of the centers to do trials. And I asked him, are you going to do these phase 2 trials? Or are we just looking at immunogenicity trials? Or are we doing placebo prospective controlled studies? And he wasn't sure of the details yet, but ask him. So let me just say that, first of all, we should have a vaccine for children. I mean, children can die of this virus. I mean, the number of children who died of this virus last year was about the same number as children who die of flu. So we consider that also worthy of preventing in children. Plus, this virus can cause, the so-called multi-inflammatory syndrome of children. We certainly have a handful of children in our hospital. And so it is, if we can prevent this vaccine safely and effectively in children, we should. But the question is how to do it. Do you want to do this as a placebo-controlled trial moving forward? Or do you want to do it as a wedge trial? Meaning that you just start to immunize children, that you do phase 2 studies and make sure that you are doing, well, do phase 1 studies to make sure you have the right dose. You do phase 2 studies to make sure that that in hundreds of children, you don't have any sort of common serious adverse event problem. And that the vaccine is consistently immunogenic with what you hope would be a predictor of protection, meaning an immunological correlate of protection. And then you could do it either as a placebo-controlled trial or just do it as a wedge trial, meaning that you just introduce the vaccine. Not all children in the United States are going to get it at once, and then you can see to what extent, at least retrospectively, it's effective and it's safe, which is the same way we did the Ebola vaccine trials in West Africa. Those weren't placebo-controlled studies. We just rolled it out, and then you could infer efficacy sort of retrospectively. So I don't know. I don't know how it's going to. I'd be very surprised if it was placebo-controlled studies. I'd see it as sort of phase 1, phase 2, and then wedge.
All right, next question to Adriana.
Hi, thank you so much for joining us today. Sorry, people keep pinging me. Okay. So we have 2 very efficacious vaccines now. So what does the speed of mRNA vaccines and their efficacy tell us about their future for preventing disease after the coronavirus?
Yeah, so I just want to sound a note of caution. I mean, the only thing we've seen so far are press releases. I really would like to have seen either a sort of rapid publication in a scientific or medical journal, so you could really drill down on the details. We don't have that yet, so let's hold off for a second. I just would like to see those data first. When we say it's 95% effective for whom and for how long? But in terms of what mRNA vaccines will mean for other pathogens, obviously people are already starting to talk about this. Will this be something we can do to create a universal flu vaccine? Will this be something we can do for an HIV vaccine? Tuberculosis vaccine, malaria vaccines, the vaccines we either don't have or vaccines that we have that don't work very well. And I'm sure they'll be tried. We'll see. I don't know. The same thing with the vectored vaccines, either replication competent or replication defective vectored vaccines. Will these sort of genetic plug and play vaccines play for other pathogens? We'll see. All pathogens are different. So I don't know. I'm open-minded though.
All right, next question goes to Taryn.
Hi there, Taryn Mento at the Public Media Station in San Diego. A couple of the other fellows took some of my questions, but I did want to ask, in terms of the priority of who it would go to first, I know that there's the CDC playbook and California has a playbook on all of the populations they're identifying, but there's a difference between who we identify should get it and how we will actually get it to them. In terms of, some people are tweeting me, is it going to be a lottery? Or are you just going to have to kind of like show up and hope you get an appointment? Kind of how are we going to close that gap between who should and who actually will? How's that decision making going to work?
Yeah, I'm not sure I'm the best person to answer that question. I'm just a scientist, but I think the people to answer that question are people like Rachel Levine, who's our Secretary of Health in the Commonwealth of Pennsylvania. I mean, I'm on a conference call with her every 2 weeks, and it's everybody from across the state, and that's the conference. That's the conversation. How are we going to do this, and who's going to do it, and how are we going to identify who to do it? How are we going to make sure they get back? I mean, for something like Pfizer's vaccine, which, is nightmarish in terms of being able to give it. I mean, could you really give Pfizer's vaccine given its requirements in a retail pharmacy? And so those are discussions we have. She's great. I don't know if you ever talked to Rachel Levine, but tell her you've talked to me and that I said she should talk to you. But I think those are the kind of people to talk to.
Is there any, can you point back to any previous experience? I know a lot about this is unprecedented, but can you point back to any previous rollout where that may give us hints or peeks at what's to come?
I think it's the problem with Pfizer's vaccine, and I'm choosing to believe this is a short-term problem, because the problem is the complex lipid delivery system. It has nothing to do with the mRNA itself. I mean, the mRNAs aren't exactly identical. They have different nucleoside analogs. One has a pre-S region, the other one doesn't. But it's the complex lipid delivery system that stabilizes that molecule. And so, whereas Moderna's delivery system is just different, it's better than BioNTech's. And I think BioNTech, just in the last 24 hours, correct me if I'm wrong, actually stepped forward and said they think they can make a much better delivery system. So this isn't going to be a problem, but it's a problem. I mean, you have, aside from shipping and storing in dry ice, once thawed, you have a day in the refrigerator, and once reconstituted, because these are coming as concentrated 5 and 10 dose multi-dose vials, you have 6 hours. So what Dr. Levine's talking about in our Commonwealth is, lining people up. We're going to do 1,000 people a day. We're going to establish centers where this is going to be done. What she wants from the federal government is she doesn't want a strategy for how to do it. She figures she can figure that out with her in her Commonwealth. What she wants is to know how much is she getting and when, and if she's going to be establishing these centers to give vaccines. I mean, in much the way you would have, say, testing centers, she needs some support for that. That's where she's coming from.
Thank you.
Okay, the next question is from Hannah Weinberger of KCTS9 PBS in Seattle. She's in a noisy place and can't ask the question, so I'm going to ask it. This is a bit beyond EUA, but how reasonably can states and counties plan for vaccine distribution and administration at this point in time? What would be impossible for states to have accounted for in their draft plans that were due last month?
Again, I'm just not the best person for that question because it's too practical.
Okay.
I don't know. I mean, I think it's going to be an enormous challenge. You have to make sure that the people who are getting it are truly first line responders, because I'm sure there are going to be people who are not sort of first tier recipients who are going to want to get the vaccine anyway and may not be honest about that. You have to make sure they come back a month later to get that second dose. I mean, I'm optimistic about that. And that if you look at Shingrix, which is probably the perfect analogy, that's a 2-dose vaccine given to adults. So the question is how good are adults coming back for a second dose? And very good, sort of in the 90% range. So that's good. And then, it's a matter of, just making sure it's in places that can give it. I asked our pharmacist, the head of the pharmacy division at Children's Hospital of Philadelphia, do you think the Pfizer vaccine could be given in a retail pharmacy? She said, absolutely not. So the only way I can imagine that happening is if there are people dedicated to just doing that. And the pharmacy would have to line up people. If you have a 10-dose vial that's only good for 6 hours after you've reconstituted, you can't just wait for people to come in. It would be wasteful.
All right, next question is going to be to Sony. Umair, your hand was up a while ago. Now I don't see it up. If you still want to ask a question, let me know. But we're now going to go to Sony.
Hi, Dr. Offit. Thanks so much for doing these. Really appreciate it. We talked a little bit about pediatric trials, and I know this is obviously one of your areas of expertise. There's still some questions about young children and how those trials will be conducted, but there are children 12 and older in the Pfizer trial. And I'm wondering if you have any insight on how older children, high school age children, will be tested. Are they just going to be rolled into these phase 3s? And how will we, how and when will we get a read on, the vaccine's safety and effectiveness in that age group?
Well, so I think if Moderna and Pfizer get approved soon, I think that's going to change the way this is. I don't think people are then just going to be rolling younger children into the phase 3 data as a placebo-controlled study. I don't imagine that. And so we'll see. I would imagine it as, first of all, how young do you want to go down, and do you want to do this as just sort of phase 1, phase 2, and then go to wedge, which is what I would imagine, or do you want to do a placebo control, which I can't imagine, and we'll see. But I would like to point out that I do think we need a pediatric vaccine. I do. Children, I mean, just round in our hospital, which I have to do in a couple of weeks, and you'll understand why you need a vaccine for children.
I guess I'm trying to kind of ask one of those questions that we get a lot. All the parents asking, when is there going to be a vaccine for my kid?
Yeah, no, good question. Don't know.
Fair enough.
I'm sorry. I wish I had more information for you. But ask Jeff Gerber. Jeff Gerber, obviously at our hospital, he's great. He's brilliant. He clearly is now on the inside of these pediatric trials, even though he couldn't answer my question about whether these are going to be phase 2 or phase 3. But he will know that. I think he should know that. Can I just make one statement? It's just as someone that's the kind of thing older people say. But when you see, for example, 11 cases of severe disease in one of these mRNA trials, and it was all in the placebo group, initially what we say when we see that is we say, great, this vaccine works to prevent severe disease. But 11 people suffered severe disease that were willing to be in these trials, and I give them credit for that. I don't know whether any of them died. They didn't say that, so I assume that didn't happen. But I'm sure at least one or more of those people were in the intensive care unit because they happened to get placebo. And I am of an age where I will now give you one rant because this has haunted me ever since I first started writing about it. The polio trials that were done in the mid 1950s, that was a prospective placebo-controlled trial. That was a phase 3 trial. 420,000 children got Jonas Salk's vaccine, 200,000 got placebo. Jonas Salk didn't want that trial to be done. He didn't. He had tested the vaccine in 700 children in the Pittsburgh area. It induced an excellent immune response. It was safe. He could not imagine giving 200,000 children physiological saline and then letting a year go by. He strenuously objected against that. So the trial was finished, and Thomas Francis stood up on the podium at Rackham Hall and said, safe, potent, and effective. Those were his 3 words. And those 3 words were the headlines of every newspaper in this country. Jonas Salk's vaccine is safe, potent, and effective. Well, how did he know it was effective? He knew it was effective because 16 children died of polio in that study. All of them were in the placebo group. 36 children were either put in iron lungs or permanently paralyzed. 34 of those 36 were in the placebo group. Those were first and second graders in the 1950s. I was the first and second grader in the 1950s. Those people could have grown up to live long, healthy, and happy lives, but didn't, but for the flip of a coin. That's the price of knowledge. And that's true always. There is a price to be paid for knowledge. And I don't think we appreciate it. I don't think we appreciated it then. We were so busy celebrating that vaccine. I don't think we really appreciated just what it took to do that. They were these sort of gentle souls we left behind.
All right, next question to David Lim.
Hi, David Lim with Politico here. I believe you've spoken to a few of my colleagues, Dr. Offit. I wanted to ask about the potential for waste in the Pfizer vaccine if it is eventually authorized. And you kind of touched on this earlier, but the limitations on how many vials can be taken out, the limitation on time once you do reconstitute it, especially in rural areas where you may not have an easy delineation of 5 individuals who might want to get vaccinated at a certain point in time, or guaranteeing that they can all be at the site at a given time. How concerned are you, if that is the potential vaccine, about the difficulties organizing that?
Very concerned. I think this is going to be really hard. I think there's going to be a steep learning curve here as various sites that are distributing these vaccines are going to be faced with these challenges, because you don't want, there's not we have 300 million people in the United States. We have 150 million Americans actually who fall into those categories of either over 65, essential worker, various health problems. I mean, that's 150 million people. That's 300 million doses. We're not going to have 300 million doses for a while. So it's a limited edition product. No, I think there's going to be a lot of waste. And I think we're going to figure this out over time. There's going to be a lot of stumbling with this. I'm sure that's true. I'm sure we're going to learn things a year from now that we wish we knew now.
In terms of the waste, do you have any sense, is there planning being done about reporting of doses that are expired, or?
That I don't know. I mean, expired, you know, so you get 6 hours once reconstituted. That's unprecedented.
All right, next question goes to Gwen Hogan, one of our 2019 fellows. Gwen, introduce yourself to the group.
Hi, how's it going? Can you hear me?
Yep.
My name is Gwen. I work for WNYC and the website Gothamist in New York. Sorry, I'm just going to a quieter room. Hey, Dr. Offit, I think we talked about measles many moons ago. It was a simpler time. My question for you, I don't know, I know that you aren't commenting a lot on distribution, but here in New York, you know, the governor has questioned, you know, the vaccine approval process, and has set up his own panel to independently look at the safety and efficacy of the vaccine. And I'm curious, sort of.
Gwen, I lost you there for a minute.
Gwen,
I think
we lost you.
You're muted again.
Sorry, I was muted. Was I muted the whole time?
No, just the last couple sentences.
So I'm curious if what Governor Cuomo has done by setting up his own independent panel to evaluate the vaccine, if you think that will overall, you know, how that will impact public trust in the vaccine, and if you think that that creates more hurdles than are necessary to get public buy-in.
Right. I definitely think it creates more hurdles than are necessary. I think what it does is it tells you that he doesn't trust the Food and Drug Administration. That's what it says, because the Food and Drug Administration will review these data. They will have an opinion about whether they think it should be approved. They'll then turn to the vaccine, the Vaccine and Related Biologicals Product Approval Committee, and we will also look at all these data carefully. We'll make a recommendation based on whether we think we would take these vaccines ourselves or give them to our family before we recommend them for other people's families. I think I understand where it comes from. It comes from what we just went through with hydroxychloroquine, what we just went through with convalescent plasma, to some extent what we just went through with remdesivir, which also is not the product one would have hoped. John Cohn wrote an excellent piece actually in Science about that. And so I can see where the distrust comes from, but I think it's time to put that aside. I really do. The everything that's in place now is what should be in place. And by doing this, by having I think at least 5 states, I was on this I had to do grand rounds at UCSF recently and yesterday. And California also has its own advisory. What will that mean? I mean, suppose that there's a couple pretty cynical people in the advisory committee who say, you know, I don't think we should get this vaccine. So you have states that are giving the vaccine and states that aren't giving the vaccine. I mean, or states that are giving the vaccine to one group but not another, and then other states that are giving it to different groups. I just think it's chaotic and it's unnecessary. I understand where the sentiment, but I just think it no longer applies. I mean, when the pharmaceutical companies get together and write a letter that says, don't worry, you can trust us. We're not going to put anything out there that's not safe or effective. And what they're essentially saying is, we know that you don't trust the FDA, but you can trust us. I think if we get to the point in this country where we don't trust the FDA, we're in trouble. You know, the FDA has successfully stood between the American public and pharmaceutical companies for, you know, since I think 1927. I mean, there's a historian named Michael Harris who says it best, quote, "The history of drug regulation in the United States is built on tombstones." I mean, every major drug regulation is based on a pharmaceutical company disaster. You need the FDA. And if you ever spend any time with people in the FDA, you know, people like Marion Gruber or Phil Krause or Doran Fink or any of the other people that are there, you'd be pretty impressed by how dedicated they are. It's too bad that this administration has successfully perturbed the view of these science-based agencies like the CDC or the EPA or now the FDA, because it has basically bent those agencies to its will. But I think that's over. I think we're done. So I think it's wrong.
Can I just do a quick follow-up on that? I'm curious, like, what you think the proper messaging at the state and local level should be for communities to start getting ready to get folks to buy in to accept the vaccine. Like, how should they be crafting that message?
I think we have to be transparent about what we know and what we don't know. Here's what we know. We know that we've tested this in, let's say, theoretically 20,000 people safely. We know that it doesn't have an uncommon serious side effect. We know that it's been tested in a variety of racial and ethnic minorities. We know it's been tested in people over 65. We know it's been tested in people who have certain medical conditions. We know that it's effective for a couple months. We don't know whether it's effective for longer than that. We think it will be, but we'll look. Worst comes to worst, we'll give a booster dose later. We know that it's safe in 20,000 people, but we don't know whether it doesn't cause, it might cause a rare side effect in 20 million people. But we're looking. Here's the mechanisms we have to look. We're open-minded to the fact that we don't know everything. But when you release a vaccine, it's not when you know everything. It's because you never know everything. The question is, when do you know enough? Do you know enough to say that the safety and efficacy of this vaccine is something that should be used now to challenge a virus, which has brought us to our knees? Do we know enough to say that? And I think we will know enough to say that. But it's not perfect. And when these vaccines roll out, I can promise you there will be people who will get the vaccine, and then a month or two later we'll get the disease anyway, and the disease might be enough to put them in the hospital because no vaccine is 100% effective. And there will be people who will get the vaccine and then will 2 days later have a stroke or a heart attack or anything else that happens in life, because the SARS-CoV-2 vaccines will be designed to prevent SARS-CoV-2, not everything else that happens in life. And so you're always going to get those temporal associations. So the CDC is going to be up against it to constantly do epidemiological studies to show that these are coincidental and not causal associations. And there are going to be pregnant women who get this vaccine, because that always happens, because people get pregnant, they don't know it yet when they get the vaccine, and then they find out they're pregnant. But there's that V-Safe system that's being run by CDC to follow up with those women to make sure that there's not a problem either with them or with their infants.
All right, an update on time here. It's 10 minutes before 2. Are you, Dr. Offit, are you able to maybe stick past to until 2:10?
I am. There may be a knock at the door, but I'll just open the door and then come back.
Okay, and it looks like I think I have 3 first-time questioners and 3 follow-up questioners. So kind of think in terms of 2-minute answers, and we'll get through this by 2:10 or 2:15. So we will go first to Leslie.
I know you said you didn't like TV right at the top, but on radio. So I'm wondering if you'll indulge me in just a I'm sure you've gotten this question a bunch, and I get this question a bunch, of sort of what are the ways that what are some of the corners that the process, what corners are we cutting, or are we cutting any corners to bring this thing forward?
I think the real answer is no. We just speeded up the process. I mean, we speeded up the process because the federal government was willing to take the risk out of it for pharmaceutical companies. The federal government said, it's basically put $24 billion into this. If you count the whole warp speed effort, that's a $24 billion effort. So hats off to Congress for their willingness to spend this kind of money. They said, look, we'll pay for phase 3 trials, even though that costs hundreds of millions of dollars. We will mass produce these vaccines at risk, which costs hundreds and hundreds of millions of dollars, knowing that if the vaccines are shown not to be safe or not to be effective or both, we'll throw them away. I mean, no company would ever do that. Companies do this sequentially: phase 1, phase 2, phase 3, mass production. They don't do it all at once. So as long as you do the phase 3 trial, that's the same standard that any vaccine is held to. The only difference is length of follow-up in terms of efficacy. That's the big difference. So we'll see. But no, I don't feel that critical. I mean, these trials of 44,000 people, that's a huge trial. That's a proof. It's a proof that you have efficacy for a certain length of time and safety at a certain level. So no, I don't think critical corners are being cut.
In one quick question in the follow-up, since the only potential thing we don't know is the long term, is there an example that we can look back to be like, oh, we did learn from so-and-so vaccine when it was done a year later, people developed X disease?
No, I can't think of any vaccine that was say 95% effective or so when, in the short term, and then was 20% effective a year later. I can't think of an example of that. There might be one that I'm not thinking of, but nothing that comes to mind. It all relates to the frequency of memory B and T cells. because that's the safety net. I mean, when the measles outbreaks occurred in 89 to 91 in the United States, there were like 50,000 cases of measles in that 3-year period, which necessitated essentially the second dose of measles containing vaccine. A lot of those children never had circulating antibodies against measles and were protected because they had memory.
All right, next question goes to Juliet.
Thank you for your availability, Dr. Offit. My question is, while we're waiting for Pfizer to come through, shouldn't we also be thinking more long-term about the different platforms as we start seeing what the efficacy is within, frontline individuals, people who are in hospitals or have to deal more face-to-face with people as the first group of folks who will actually get the vaccine? What do you think is likely to occur while other companies start developing their vaccines?
Julie, do you mean what is the likelihood that I think that these other sort of either replication defector or replication competent vectors will have a chance in America? Is that what you mean?
Yes, and should we be also waiting for other folks to come through with other platform types?
I see what you're saying. Waiting, no. I think if you have something that looks good, start. And if you may find that there is a safety issue that surprises you, you may find that the efficacy fades, which will surprise you negatively. But I don't think you're right. You're dead right in that the first vaccines are often not the last best vaccines. I mean, the first Haemophilus influenza B vaccine, HIV vaccine was replaced by a better vaccine 2 years later. The shingles vaccine in 2011 was replaced by a better shingles vaccine in 2017. The first measles vaccine in 1963 was replaced by a much better measles vaccine in 1968. The first rubella vaccine in 1969 was replaced by a safer rubella vaccine 10 years later. So the first vaccines are often not the last best vaccines. But I think as in every one of those cases, I think you start with the what you have in hand, and then if it ends up being safer or more effective down the road, then you do that. I think that's historically true. You can't wait for that perfect vaccine, not initially. I just don't think it. Certainly now we have the time to do it.
Thank you.
All right, next question, Mary, and tell Dr. Offit where you're from.
Mary Hines, Las Vegas Review-Journal, and I actually think this is kind of a very general, open-ended, not practical question. And I put my hand down because maybe it's a little ridiculous, but I'll ask it anyway. Just overall, I think the public wants to know how optimistic they should be. And I know piece by piece you're giving us that answer, but how optimistic are you with this vaccine? How optimistic should the public be?
We're a cynical lot. So we're always nervous when we get so optimistic that something bad's going to happen. But if you let me put it this way, if you ask scientists in January, when we first published the sequence of this virus and we had it in hand, whether they thought that 11 months later, ask 1,000 scientists in the United States, 11 months later, we would have completed essentially 2 large prospective placebo-controlled trials using a messenger RNA technology that showed protection against mild, moderate to severe disease in a 95% efficacy level, including in older people who are over 65, also over 90% effective in people over 65. I think you would have not had one scientist that would have raised their hand and said that they thought that was possible. Not one. So I think it's amazing. I mean, to be honest, I think what we're seeing is remarkable. It is a tour de force. It is a scientific tour de force. But like all of us, I guess, in this country, you're just a little nervous because the virus is new and because these strategies are new to go sort of too far overboard. But I think you should be incredibly optimistic that we now have in hand, in all likelihood, a way out of this mess.
Great. I'm glad I asked. Thank you.
Yes, great soundbite there. Next question to Lisa Krieger again.
Let me unmute. Thank you. Can you explain what boosters do and why the Moderna and Pfizer have separate, I guess, different timings on the booster? And then second question, why do vaccines elicit an immune response that's stronger than the actual infection itself? Just some geeky science questions, but important.
So in terms of doing, say, 3 months after dose one, or I'm sorry, 3 weeks after dose one, or 4 weeks after dose one, it's just the way they did the trials. I don't think there's anything magical. I think if a mistake was made, for example, and somebody got a Pfizer vaccine, and then instead of giving it 3 weeks later, was given 4 weeks later, or vice versa, I don't think that would matter. It's interesting that some vaccines induce immune responses that are better than natural infection. It's true. I mean, if you get tetanus and you have symptoms of tetanus, you still need a tetanus vaccine. The reason being that the toxin that tetanus makes, the bacteria makes, so-called tetanospasmin, can induce symptoms at sub-immunogenic levels. In other words, it takes such a little bit of tetanospasm to induce symptoms that you don't make an immune response to it. So you still need a tetanus vaccine. The conjugate pneumococcal and Haemophilus influenza B vaccines induce better immune response in babies, because babies, it's a sort of technical science, that's an immunological question, but babies don't make very good polysaccharide immune responses, that complex sugar that sits on the surface of those bacteria. They don't respond well to it. Babies who get HIB, who have Haemophilus influenza B, still don't have good immunity against Haemophilus influenza B. So they still should get the Haemophilus influenza B vaccine. So that's another example. The human papillomavirus vaccine actually induces an immune response better than that it's induced after a natural infection. Those little virus-like particles are just highly immunogenic.
Can you explain just briefly what boosters do, like scientifically, and why they're important for people to follow up?
Yeah, so you have a certain frequency of memory B and T cells that are induced after the initial vaccine. Then you get a second dose, and that boosts the frequency of those cells and boosts your antibody response. So that's what happens with boosters.
Got it.
And it just depends on the pathogen and how the vaccine is made as to whether or not you need boosters, and whether those boosters have to have adjuvants in them.
Thank you.
But the one thing, people have hoped that maybe this vaccine will be better than the vaccine induced by natural infection. But I would say this, I mean, when we were making our rotavirus vaccine, there was a paper published by Ruth Bishop that was the critical paper, because what she found was that the babies who got rotavirus infection, that when they were then exposed to the virus 2 or 3 years later, they could still be reinfected, but it was only mild infection. It wasn't moderate to severe disease. When you see that natural infection can protect against moderate to severe disease, then you know you can make a vaccine. Now, this virus has only been out for a year, but the stories you haven't heard are got moderate to severe disease, recovered, got moderate to severe disease again. You haven't really heard those stories. That's a really good sign that if natural infection can protect against disease, then I think you're going to be able to make a vaccine that also protects against disease. And that's not always true. I mean, you can get gonorrhea again and again. You can get strep throat again and again. Yeah. That's why we have that.
Quick follow-up with the memory B cells. Are we actually looking for a count like we do with antibodies? Is that what metrics?
It depends on the pathogen. I mean, you really only know what the number means with regard to each pathogen.
Okay, thank you.
Let me see, Andrew Dunn.
Dr. Offit, on a different front, what are your biggest questions, concerns on manufacturing for these AdCom meetings, information you want to get out of these to feel more comfortable? Specifically, Moderna has never made a commercial medicine. It's a new platform. For an EUA, the FDA said they're not doing pre-approval inspections, so that Norwood manufacturing facility has not been looked at by the FDA to our knowledge. Does that concern you at all? And what do you want to see as far as actually what can convince you they can actually make 1 billion doses at GMP level?
Right. So the FDA's phrase for this is CMC, chemistry, manufacturing control. If you looked at, I don't know if you saw the VRBPAC meeting on October 22nd, but there was one word on one of those slides that was presented, it may have been by Phil Krause, but it was, the word was generally, when they were talking about how they inspect the building, et cetera, that was generally held to the same standard. When I watched Merck make its vaccine against rotavirus, the FDA asked, they don't just license the product, they also license the process and they license the building. In the licensing of the process, it's really rigorous. I mean, they have you validate every aspect of the process that makes the vaccine, because, for these products, which are biologicals, the process is the product. I mean, it's not like a small molecule drug like amoxicillin where you can say, yeah, I got 50 milligrams of amoxicillin in this tablet and 50 milligrams in this tablet that's made with the next dose. It's a biological. The process becomes the product. And it's really rigorous. And it took a year. I mean, it took a year for Merck to just validate their process. That's obviously not happening here. Is there a critical loss of supervision when they do stand by? Now, they've been involved since the beginning of the FDA, but not in the same manner that they would have been for a typical licensed product. Does that matter? Great question, and I'm sure that question will be asked. Andrew, did that answer your question?
Yeah, that's a corner cut though, right? Like we should call a spade a spade. Like this is a normal FDA, this is the urgency of the pandemic, but normally you would have a pre-approval inspection of a plant and they're not doing that. And the consistency lots, as far as those 3 different lots, they're not necessarily requiring that either, correct?
No, you're right. And so then the question becomes, and similarly, you don't have the kind of efficacy follow-up that you would like to see. So the question is it critical? Does the fact that that's not happening make for a less safe product or a less potent product or a less consistently made product? That's the question. And the answer may be no, but I think, it's because when people hear the term corner cut, they assume there's been a negative outcome. That's not necessarily true.
All right.
I don't think I have a lot more time actually, Chris, just because I have to do this next thing.
Okay, actually, we're on the last question. You got time for one more?
One question, sounds good.
Okay, Chair Mento, last question to you.
Yeah, so I know that we've been testing this in people with a variety of medical conditions. I'm wondering if those are only physical medical conditions, or if mental conditions are also considered, and if there's any concern that efficacy or consequences could arise from individuals who may have depression or substance abuse at all.
I haven't heard any mention of that.
That the people with mental health are being conditions are being considered as a medical condition in these trials, or either.
I hadn't heard that that was being considered. It was mostly sort of focused on obesity, chronic obstructive pulmonary disease, diabetes, those kind of heart disease.
I'm sorry, I have one more. If we can take this from Anna Sussman. She was on the list and I forgot
to ask. You lied to me. No, I'm sorry.
I did. Anna?
Hi, thank you so much, Dr. Offit. Question really quickly, the different candidates, I was wondering, based on their formulations, if you had any thoughts on which one might be most suitable for pregnant people, when you expect to have some of the data you mentioned, and also based on what you know, like where you think pregnant people should fall in this sort of prioritization.
Yeah, these aren't live products. So I don't, and number one. Number two is I don't think that they given their structure, that they would likely cross the placenta into the child circulation either. But there was somebody at the door. But so I don't know. I don't think so. Certainly there will be pregnant women who will receive these vaccines, who have already received these vaccines, and there will be follow-up through the so-called V-Safe system. I don't know if you heard the CDC talk about that, but they will follow that up. But again, I don't know is the real answer, because you're always surprised by these things, but I don't anticipate a problem.